I want to welcome everybody to our second annual Musculoskeletal Infection Research Interest Group. I'm Noreen Hickok, and I'm one of the co-organizers, along with Amber Jennings and Antonia Chin. And we think we have a phenomenal program today, which should touch on many aspects that we're so interested in. So I have to do some housekeeping things first, which is to remind you that you're being recorded, that all participants are muted upon entry, and we ask that you stay muted, please. Equally, we're going to ask you to leave your cameras on for the first couple minutes because the ORS is going to take pictures so that we can put together a participant list for this RIG. We had a picture in our first one, which we've included in a document that I've put into the chat. Now, that document in the chat's important because we're going to use our time during this to have the speakers speak as long as possible. And so we're going to have the discussion session in a separate Zoom link that's included in that document that I've put there. It gives you the order of speakers as well as the Zoom link, the picture from last year for those of you who were here, and also who the organizers will be for next year. So I encourage you all to download that and join Amber, Antonia, and I in welcoming Ebru Oral, Tom Scher, and Corda Syed as the organizers for the third RIG. So we have to ask you to turn off your video after the first couple minutes so we can preserve bandwidth. And please do not take screenshots. That's forbidden. And only with the permission of both the ORS and the speaker is that allowed. And the ORS asks that you take a minute to enter your name in the chat box and reserve that for questions. They will be sending us the questions to take them over to our separate Zoom session. And they also ask that you consider becoming a member of the ORS if you aren't already. So with that being said, I am very pleased to welcome our first speaker, who's David Granger. Now, I feel incredibly honored that we have Dave speaking with us. He's a University Distinguished Professor and Department Chair of Biomedical Engineering and Distinguished Professor of Pharmaceutics and Pharmaceutical Chemistry at the University of Utah. Dave has an incredible list of honors, boards he serves on, et cetera, and frankly, we don't have time to go through them, but I am really looking forward to his talk. So Dave, you are on.
Thank you very much, Noreen. What a delight to be here. Thanks to everybody for attending. And I will move directly here to share my screen and start my talk. So I thank the ORS and the organizers for thinking of me, and I'm honored to participate. So here's my conflicts. They're also posted on the ORS site. So let's start here. The implant host response, of course, has been well documented in terms of what's called the foreign body response, or the FBR. And this is a scenario in which, and to my knowledge, nearly every single biomaterial implanted into both hard and soft tissue produces a series of cascades, beginning with microsecond events in the protein level, and particularly complement activation, and then moving through to macrophage polarization, neutrophil exhaustion, frustrated phagocytosis, and wear particle release. So here's the dogma for the foreign body response, and it's been well documented. What is less understood beyond pure phenomenological issues is, in fact, how neutrophils, as being one of the first responders to a biomaterials implant, are dysregulated. And there's a number of confounding signals and complex cascades that are involved in that. Equally confounding is what macrophages do in a different temporal sequence after neutrophil invasion. And the classification of macrophages, of course, I think is under some redefinition. How they respond to the myriad signals in that site is also equally confounding. And so what is well known, as shown here on the upper right, is that eventually 3 weeks, 3 months, depending on the site of implantation and the exuberance of the response, is that these implants become encapsulated with a avascular collagen-like matrix, and we characterize that as the fibrous capsule. More recently, I think that the interesting connection between implant infection And this dysfunction in the normal healing cascade has been, I think, actively investigated. And it becomes then an intriguing question as to what those types of relationships might be, where the dysfunction in our normal wound healing process around an implant leads to, in fact, an enabling environment where the implant is a nidus for infection. So the mechanism of this, despite the phenomenological aspects, is quite speculative. And that's because it's quite complicated in involving so many different cell types, so many different temporal features, patient factors as well, and including even surgical and perioperative factors as well. So you might be able to circle on this diagram that diagrams the typical foreign body response and the myriad of signals and cell bodies in there. Pro-inflammatory features, anti-inflammatory features, pro-regenerative features, anti-regenerative features, immune stimulating features, non-immune stimulating, immune suppressing features. It's all there in the FBR. And so now if we want to tease mechanistic features out that in fact produce a pathway towards susceptibility to infection, susceptibility to pathogen colonization that leads to infection, any of these early mechanisms and the outcomes of infection in implants, it's quite difficult to point to any of these signals as being key indicators for doing so. From the pathogen side, pathogens are well known to also undergo immune evasion at the implant site. So here's a nice Italian figure that I like a lot, in which the biomaterial, with obviously the cartoon of a Staph aureus biofilm there, now produces an enormous amount of different signaling pathways, and all of these pathways are directed at the same actors that are involved in the foreign body response, that is the macrophage population and the PMNs that are there in presumably an inflammatory, an immune communicative pathway, and yet they are acted upon by a myriad signals now from the pathogen side. So the pathogens essentially reprogram the local environment around the implant that changes the host immune response, the inflammatory cell behaviors, their phenotypes, their ability to respond. And so this induces then a mechanism of ostensible tolerance at the implant site and a frustration in terms of the inflammatory cascades proceeding towards normal wound healing. So infection is in fact allowed to persist. Is the host actually permissive in a participant in doing this? And you might say, well, it's a willing or unwilling participant, depending upon the types of signaling involved. So we have now the host signaling that's confounding to the implant. We have the host signaling that's confounding and enabling to the pathogens. And now the pathogen signaling that's also quite controlling to our inflammatory and immune response. So we know from tumors, the tumors are well recognized in their intrinsic ability to foster host-tolerant environments. And so we know that tumors produce these immune suppressive signals, and that has led to more recently an enormous amount of cell-based and pharmacological-based therapies called checkpoint inhibitors that are useful for reprogramming the tumor environment. So the question is, can we take learnings from that and now apply it to the implant local tumor tissue immunosuppressed environment? And we have this analogous situation to tumors where there is an immunosuppressed environment and host tolerance to those same responses. Is there, in fact, an analogous parallel? The host infection tolerance is induced by different types of signals. So pattern receptors that cue up the toll-like receptors on our cell bodies, and then also damage-associated, pathogen-associated molecular patterns that are well known from pathogens and trauma signals that are keys to controlling the host immune response. So the immunosuppressive environment is a little bit different in the implant scenario and infection that it might be with the tumor, but the concept is similar. Pathogens exploit then this immunosuppression, and their ubiquitous colonization induces host tolerance. There are several elegant strategies for doing that, and I think that some of these will be developed here by speakers later in the session. So the pathogenic microbes have this sophisticated armada of strategies to subvert the host defenses and even potentiate the foreign body response. And so there's this signaling back and forth. And the question is, can biomaterials even change this? So the implant site has then a number of myeloid-derived suppressor cells, T regulatory cells, macrophages, and neutrophils that become dysfunctional. There's immunosuppressive cells that secrete anti-inflammatory cascades to keep it immune suppressed through the action of ROS and RNS species. And so things that might be pro-regenerative in a normal wound healing context actually now, like TGF-beta and IL-10, become immunosuppressive actors at an implant site. Immunosuppressive cells that result then suppress other effector immune cell functions through the actions of arginase 1 and IDO activation. I think this is well established in the immune suppressed environment. And then these bystander effects go on to produce cell senescence, fibrosis, osteoporosis, atrophy, and degeneration of the extracellular matrix. So the impaired homeostasis, the result of all these signals then exhausts the neutrophils, alters the macrophage phenotypes, induces signaling cascades that produce fibrotic tissue instead of normal tissue suppression, normal tissue generation. So immune suppression prevents inflammatory damage in trying to downregulate these inflammatory processes, but then the consequence is that it promotes tissue degeneration, enabling infection. So we know that the infection is cleared once an implant is resorbed. There's numerous stories about how sutures go away and fibrosis goes away, how different types of degradable polymeric implants, resorbable calcium implants, once they resorb, all of these features revert back to normal wound healing. But as we know, many implants don't resorb by design. So tissue healing, homeostasis, inflammatory balance, and immune competence, we'd like to be able to restore them once the foreign body is removed. And we see this happening under the case of resorption. And the question is then, can it be then mimicked in the case of a permanent biomaterial? Can a permanent biomaterial instruct the tissue site away from these adverse implant and pathogen steering mechanisms and back towards a normal healing homeostatic phenomena? And of course, this is not a new idea. Buddy Ratner talked about biomaterials that heal back in 2007. And so the idea that this is now, 15 years later, and we really haven't developed this idea very significantly towards any clinical fruition beyond some, I think, rather simplistic, instructive, but in simplistic academic studies. So the design criteria to create biomaterials that heal, we don't have design criteria, in my opinion. Active responses to implant environments that actually actively restore inflammatory and immune balance, I would argue that we aren't there yet. And so we have this quote that's attributed to Einstein, but actually it's not attributed to Einstein. That's a misnomer. But nonetheless, we have this problem of going down the same pathways that we always go down, releasing a single cytokine, releasing a single anti-inflammatory NSAID, releasing a single cell pro-regenerative cytokine. These processes, we've done this for 2 decades now, and we really haven't produced a major impact in immunomodulation. So we publish hundreds of studies annually describing antimicrobial technologies for medical devices. So why don't we see then this being clinically approved? So we've got then non-predictive models of human implant infections, inconsistent results, a lot of overly complex implant solutions that are proposed that will never make clinical trials because there are just too many special sauces and secrets and trade secrets and other things mixed into a single implant, multiple drugs. Even releasing 2 drugs at the same time is a very, very difficult thing to get through a regulatory pathway. So we have few mechanistic studies that instruct us about how to create biomaterials to immunomodulate or even create an anti-inflammatory scenario that's clinically reliable. So this erodes our credibility as an academic environment in that we have empty academic promises with thousands of papers annually, but relatively little moving towards any clinical trials beyond a mouse model. So we have implant material roles that we recognize. We have in vitro studies that show biomaterials can control bacterial adhesion. We've got lots of in vitro studies. It can modulate immune activation in single cell cultures. We've got lots of studies that facilitate bacterial phagocytosis from surfaces by reducing interfacial forces and adhesion in vitro studies. We can kill at a distance using released antimicrobials. We can kill locally releasing antimicrobials. We can use contact surface killing of immobilized agents to kill pathogens. But we haven't solved a lot of the serious problems and that need to be addressed in vivo. And so far we've learned, I believe, that implant materials alone cannot yet eliminate the foreign body response or the endpoints of the foreign body response that perhaps are the nidus and enabling to infection and pathogen colonization. So Sorry, that's my timer. So we can eliminate the foreign body response. So we can't eliminate that. We can't prevent perioperative contamination. Remember, this is a systemic holistic problem where every operating room is contaminated. We've got lots of documentation to show lots of ways that the patient's contaminated. Sometimes the implants are contaminated, the surgical site's contaminated. So that seems to be a systemic problem that's separate than the implant biomaterial. We are not able clinically to prevent biofilm formation. One could argue that that is perhaps the center of the problem. We are not able to prevent host suppression or neutrophil exhaustion. I think that's clinically recognized that biomaterials implants consistently produce this immune suppressed environment. We can't address persister cells in vivo very correctly. We can't deal with antibiotic resistance in the context of antimicrobial adjuncts to our implants. We're not able to effectively promote tissue integration that prevents bacterial colonization. That is, the race for the surface is still a race for the surface. And it's an interesting hypothesis, but we practically haven't been able to do that in vivo. We're not able to eliminate intracellular pathogens. It's well known that osteoblasts and macrophages can be colonized with the numerous pathogens, notably Staph aureus, and they produce small colony variants, which are equally problematic as slow metabolizing, slow growth variants of clinical pathogens. And so we're not able also through the foreign body response to restore normal tissue homeostasis. So let me finish up here. This idea of restoring immune competence to protect implanted sites is a balance then of the antimicrobial nature of keeping pathogens out and the pro-healing nature of providing a wound healing environment that's endogenous to the host. So balance is key, and the balance between the host immune response and the inflammatory response in the context of implanted biomaterial. So the modulation strategies can be immunomodulatory using pharmacology or biomaterials, or anti-inflammatory using pharmacology or biomaterials. But we have to be careful of the balance, because an imbalanced immune response by provoking an immune modulatory proactive biomaterial could lead to adverse events in an imbalanced immune response that is now hyperactive at the implant site. A well-intended anti-inflammatory approach could also be, and then also a wrong approach, where we need to think about the balance of what's appropriate. As we eliminate these responses, we might make the site more fibrotic, more reactive, less able to clear pathogens. So infection mitigation and healing is a tough pathway. If we're going to stratify the response into these two dichotomies, we have to worry about how we're going to control that in situ. So the idea of revisiting then this checkpoint inhibitor, or how CAR T therapies work and tumor immunotherapies, right? This is work in progress, but it's an interesting hypothesis about how we're trying to reawaken the tumor site to recognize the potential to clear the tumor using the proper immunomodulatory balance and restore the stasis in the tumor site to clear that disease. And that approach could also, I think, be very useful to think about in the context of implants. So I'm going to focus at this. Sorry, I got too many slides here. So let me just clear up here. The biomaterials confound the host response, and the inflammatory responses compromise implants to pathological invasion and to chronic fibrosis. Bacteria use then several strategies to produce a problem in the implant site, such as invasion, toxin production, and skewing, active skewing of the local immune response. Antimicrobial experiments are compromised by pathogen strain selection and animal model limitations. This is a fundamental experimental issue, which I don't have time to talk about, but I hope comes back in the discussion. And biomaterials may alone not be able to exercise the complexity, the sophistication, the potency to be able to deal with this whole thing holistically. The immunomodulatory capacity, antimicrobial capacity, tissue regenerative capacity, and their functional capacity as a prosthetic. Addressing then these defective immune circuits in the implant tissue environment with implant strategies is opportune. There's a number of things from tumors we can learn, and I think we'll look forward to taking it from there. Thank you very much.
Thank you very much for a fantastic presentation. We could listen to this for hours and hours, and that's why we thankfully have a discussion section afterwards. So please all stay on afterwards, and we'll put that link up there afterwards to make sure that we can still continue discussion. So thank you for that presentation. Next, we'll have Bing-Yin Lee talk about new biomaterials on the horizon for orthopedic infection. He's a professor of orthopedics at West Virginia University and a huge advocate for musculoskeletal infections. So thank you for leading this section for musculoskeletal infection, and we look forward to your talk.
First, I would thank the organizers for organizing this great week meeting, and it's an honor to present here. And I also would thank Dr. Granger to give a great talk on the challenges we all have been facing. And as always, his talk is very inspiring. Meanwhile, I'm also, as you probably are, optimistic and very glad to see the progress we all have been making in this research area. And today I will briefly give a few examples on biomaterials development. I have nothing to disclose. So as Dr. Granger mentioned, there are quite a few challenges we all have been facing related to musculoskeletal infections. This may include anabolic resistance, biofilm formation, intracellular bacterial, delayed wound healing, as well as the challenges related to diagnosis and non-invasive monitoring. I will give one example to each of these challenges. I apologize that most of the examples are nano-related because of my research background in nanomedicine, and also because of the success of nanotechnology in the development of COVID-19 vaccines. As we know, the Moderna and the Pfizer vaccines all use the lipid nanoparticles to encapsulate the messenger RNA. And the nanotechnology has unique effects by a targeting that can reduce amount and the side effects of the vaccines or other type of drugs. And also this lipid nanoparticles could have reduced nucleus degradation, and controlled release may also lead to the reduced doses. And co-delivery of vaccines and adjuvants could improve the effects of the vaccines. And these lipid nanoparticles could also be readily accessing the nearby lymph nodes, which is important for the vaccines to interact with the immune cells. So all of these features of nanotechnology have been used to develop biomaterials, as you may see in some of the examples we will talk about. So one of the challenges with a musculoskeletal infection is anabolic resistance. Pathogens have developed a resistance to all kinds of antibiotics we are using. And we prevent infection or treat infections. In general, there are two different approaches. One is chemical approach, where we use antibiotics or antimicrobial agents to kill the pathogens. The other approach is physical approach, and researchers have shown that animals like the cicada's wing surface and also the, excuse me, the dragonfly wings, as well as the gecko skins have unique nanostructures, and these nanostructures have enabled the surface anti-fouling properties or resist of bacterial colonization. As you can see in Fig 2, researchers have developed nanostructures that can have anti-fouling properties or resist bacterial colonization on both static and flow conditions. And Fig 3C shows that unique nanofido, these needles can even poke the bacterial cell walls and kill the bacteria. Another challenge is biofilm formations. One of the publications on Journal of Orthopaedic Research showed that the combination of bacteriophage and antibiotics can reduce biofilm formation and infections. So the good thing with bacteriophages is that these bacteriophages are naturally occurring viruses that have high specificity toward the bacteria, but may cause little effect on the host cells. And the downside, of course, in this area is we do not have a lot of clinical trials. And another challenge is intracellular bacteria. So in this case, intracellular bacteria are believed to contribute significantly to chronic or recurrent type of infections. Researchers have developed antibody conjugates that can target intracellular bacteria. In this case, researchers have developed conjugates of rifamycin type of antibody conjugate with MESA-specific antibody. And these conjugates can bind to the surface of the bacteria. And these conjugates and bacteria will be internalized by the host cells like macrophages and osteoblast cells. Once they are internalized into the host cells, the protease within the phagolysosome will cleave the link between the antibody and anabolic, and the cleaved antibody will be activated and eliminate intracellular bacteria. The researchers have shown that multiple or many these conjugates can bind on one bacteria, so they can have high enough antibody concentration to eliminate the intracellular bacteria. And the figure B showed that before the cleavage, you will not see the green fluorescence, which is quenched, but once you cleave the link between the antibody and the anabolic, you will be able to see the green fluorescence. And figure C showed that the clinical or the animal studies show these conjugates are effective in eliminating the intracellular bacteria. So another challenge is, as Dr. Granger mentioned, that have to balance infection and wound healing. So its infection may lead to the delayed wound healing. This is not a surprise, as we know the bacteria and human cells or mammalian cells are competing for the surface of implants, as well as the tissue surface, so-called racing for the surface. So researchers have been developing dual functional biomaterials to label the biomaterial surface have antimicrobial property, but also can promote bone regeneration or bone healing. In this case, a variety of anabolic agents have been studied, like antibiotics, antimicrobial peptides or nanoparticles, and like collagen particles, and to label antibacterial properties, to label properties that can promote bone healing. Studies have shown that incorporate magnesium, zinc, or silicon nanoparticles, and of course, growth factors like BMP2, BMP7. And again, of course, in the end, is how to balance those impact. So another challenge is how we can be fast and accurate to diagnose the infections. In this case, Dr. Schwartz's lab have developed antibodies as biomarkers for infection diagnosis and prognosis. In this case, they have extracted antibodies from antibody sequencing cells from blood samples, and they have shown that the levels of the antibodies would decrease if the clinical treatment was successful. However, if the clinical treatment failed, you would see the increase of the level of the antibodies in the approach MENSA samples. So this is a very interesting approach, and definitely, you're welcome to talk to Dr. Schwartz and his lab members to know more details of this study. Another challenge is how we can monitor the infections non-invasively in animal models. As we know, bionumerous bacteria have been developed, and more recently, researchers have developed the LUC reporter, which is better than the previous LUX model. Compared to the LUX model, this LUC reporter has better penetration of the tissues and also gives stronger signals. And by combining use of this LUC reporter and LUX reporter, they could monitor the infection progression of two different types of bacteria. So this will be more, of course, reflective of what we see in the actual infections, where you see two or more bacteria. So just in summary, I think we are happy to see the progress in developing new biomaterials dealing with musculoskeletal infections. Of course, as Dr. Granger mentioned, we do need to find ways to see how really get some products that can reduce infections in the clinics. And also we know that there are some antimicrobial agents have strong antimicrobial properties, but they have limited clinical uses, likely due to the high toxicities also toward the host cells. So is there a way by taking advantage of the technologies like nanomedicine to reformulate or redevelop this type of antimicrobial agents to make them have high antimicrobial properties and also have limited toxicity toward the human cells, so they could have a broad impact in clinical applications? So with that, I would thank the people who have worked in my lab on my projects. We have had more than 100 trainees going through the lab since 2005, and I would thank the financial support from the funding agency. And thank you for your support. I will be happy to answer any questions.
Thank you so much for a fantastic presentation. We're going to now move to Mark Smeltzer talking about a bug-eyed view of biofilm-associated orthopedic infection. He's a professor of both microbiology and immunology, as well as orthopedic surgery at the University of Arkansas. Please welcome in joining Mark.
Thank you. By way of further introduction, I am a bacteriologist. I have a specific interest in Staph aureus, and even more so in Staph aureus as an orthopedic pathogen. What I'm going to talk about today is sort of an inside-out bug's eye view. We're going to talk about the bug. So I thought I would start a little bit unconventionally with what a biofilm is not. I think everybody attending and everybody presenting knows what a biofilm is. And the only point I wanted to make with this from the bug's eye perspective is that what it is not is an accident. It isn't something that just passively accumulates on an implant or on bone or any other surface. It is highly programmed and representative of a unique lifestyle. So from the point of view of the bug's eye, this is a figure taken from a paper several years ago that was meant to clarify regulatory circuits in Staph aureus. The paper is really good, and I don't mean to disparage it, but whether or not it clarified anything, I'm unclear about, just because of the complexity, and that's reflected by all the interconnections that you can see in this paper. So the way that my lab has approached it is just to ask the direct question, starting with what a biofilm is. How does this pathway impact biofilm formation? What are the critical elements within it that do so? So here's a figure from several years ago from a MD PhD in my lab, and all it's intended to show you, we went and mutated all of the regulatory loci that we could mutate and just assayed biofilm formation. And if you look at the red arrow, you see that without question, there's one gene in particular called SAR-A, or the Staphylococcal Accessory Regulator A. There are other SAR-T, SAR-U, etc. But if you mutate SAR-A, you essentially do not form a biofilm. And that was of obvious great interest to us. Now, SAR-A is a regulatory, it's a DNA binding protein. It's not an enzyme, it's not an adhesion, it's not a toxin. It's a regulatory protein. It binds DNA and changes the way genes are expressed. So it wasn't immediately obvious why SAR-A mutants don't form a biofilm. This slide is intended to, after many trials and tribulations that we all go through to reach a relatively simple conclusion, is pointing out, I don't know how to make my pointer work. So in that top figure, wild type, that's a wild type strain, that's a zymogram. What you're looking at is protease activity. S is indicative of a SAR-A mutant, and you notice the protease activity goes way, way up. P is indicative of a wild type strain that can't make proteases. And SP is indicating a SAR-A mutant that can't make proteases. And in the bottom figure, what you see is, again, mutation of SAR-A essentially abolishes the ability of Staph aureus to form a biofilm, at least as assessed by this in vitro assay. And all you have to do if you want to not only restore it, but in fact enhance it, is to eliminate the ability to make extracellular proteases. So what it's suggesting is that the reason SAR-A doesn't form a biofilm is because of dysregulation of protease production, and consequently disruption of the biofilm. Now, biofilms get a lot of attention in orthopedic infection, particularly implant-associated infection, but we don't think it's the whole story. And if this movie will work, I think it will exemplify that. This is from a murine model where we drilled a unicortical defect in the bone, infected it with Staph aureus, and you can see pretty readily that you get all this cortical bone destruction that I don't think is associated with biofilm formation. So now I guess I have to let that movie run. So remember, while that happens, that SAR-A, mutate SAR-A, it limits biofilm formation. That's important. We think that's important. Remember also, though, that orthopedic infections involve elements that go beyond biofilm formation, that include cortical bone destruction, this reactive bone formation, all kinds of changes in the bone itself that are, we don't think, related to biofilm formation. And remember also that our data suggests that the reason a SAR-A mutant doesn't form a biofilm is that it excessively produces extracellular proteases. So what I'm showing here is from a paper we published last year that is intended to illustrate what we have found to be an inverse relationship. Doesn't really matter. And again, I apologize that I don't know how to make the pointer work.
We can see your pointer. It's actually good. You can see you can use your mouse. And you can see your pointer.
You can?
Mm-hmm.
Okay, then here's what I would, thank you for that. Here's what I'd like you to notice. This is a protease assay, and from left to right, proteases go up. This is our mouse model, where we have a unicortical defect that we infected with different strains of Staph aureus. This is the parent strain. And you notice an inverse relationship, because as proteases go up, all those changes in bone remodeling go down. So you get less cortical bone destruction and you get less reactive bone formation. So all of those things that we think are also critically important in the therapeutic recalcitrance of these infections, that being the ability to deliver an antibiotic, even if resistance isn't an issue, you still have to get it there. And we think these kind of changes compromise that. So again, SAR-A, the impact of SAR-A appears to go well beyond biofilm formation itself. This slide is intended to illustrate that, again, we're looking at this in a comprehensive fashion. This slide is comprehensive in two respects. One are the strains that are over here on the far left of each graph. These are two very, very different strains. One is methicillin resistant, one is methicillin sensitive. They are essentially unrelated to each other, as two Staph aureus strains can be, but they share the common feature that if you mutate SAR-A, protease production goes up, and it goes up much further, to a much higher level than any of the other regulatory mutants that we've looked at. So in the interest of time, I just put this rather busy slide together, but I included, knowing this would be recorded and the attendees would have access to it, critical references from my lab, that mutation of SAR-A we've demonstrated increases protease production more than any other Staph aureus regulatory locus we've ever looked at. Conversely, it limits biofilm formation more than any other regulatory locus we've looked at. That third bullet point's really important, because it's easy to show that you can get a statistically significant decrease in biofilm formation, but the reason biofilms are a problem is because they confer anabolic resistance, intrinsic anabolic resistance. So I'm more concerned about the fact that mutating SAR-A increases anabolic susceptibility than I am about any statistical significance you might read in the literature. I would also note that we've found regulatory genes that have the opposite effect of SAR-A. But importantly, if we mutate those with SAR-A, we can completely reverse that effect, meaning it doesn't matter what the status of any other gene is. If you mutate SAR-A, you can't make a biofilm and you don't cause that bone destruction. We've also shown that that's associated with limited cytotoxicity for osteoblasts and osteoclasts, and as I just illustrated, limited virulence in an osteomyelitis model. And then that last bullet point, again, I think most people don't understand. They talk about Staph aureus like it's a thing. It's a singular thing, and it is not. There are very different strains of Staph aureus, and that's reflected primarily in anabolic resistance, but it goes far beyond that. So I think it's important for me to note to the group that all of these things that are important in orthopedic infection, if mutation of SARA does all of them in every isolate we've ever looked at. So the question becomes why? So you got SARA, you mutate SARA, proteases go up. What are the proteases doing? Well, this is a recent, one figure from a recent paper with one of my doctoral students showing that basically what happens is the entire virulence factor repertoire of Staph aureus gets remodeled by these proteases. If you don't control their production, essentially Staph aureus eats itself. So you can look over here, for instance, all of these Staph aureus proteins, fibronectin binding proteins, coagulase, some we don't know anything about, are present at least 32-fold higher concentration in virulent strains in our murine model by comparison to attenuated strains. And in the interest of clarity, that was done with this set of strains. So those proteins are all present 32-fold higher in these, both of these, by comparison to all 4 of these. So we think that suggests a cause and effect, or a potential cause and effect relationship that still needs to be investigated. And then finally, and you'll have to forgive me, please, because I actually just got this data yesterday, and Noreen knows that because I was sending her slides this morning, I was a little bit conflicted whether to go this route versus another route. But what we're looking at here, so what I just showed you in this slide, SARA represses protease production, and the failure to do so degrades Staph aureus virulence factors that are important in the pathology of bone infection and biofilm formation. That's a take-home message up to this point. But then the question, we turned our attention to, well, what happens in the bone? So here we just took infected bones and made homogenates. This is bone, all its associated tissues. We did not rinse out the medullary tissues. We just made homogenates, and we did global proteomics. So here in the Venn diagram, you can see that we identified 119 proteins, host proteins, that are altered up or down in a significant fashion in Staph aureus infected bone versus uninfected bone. And then importantly to our focus on SARA, and that, sorry about that timer, all but 13 of those are restored to their normal levels in a SARA mutant. You can see that perhaps a little clearer presentation over here, that if you just look at all the protein profiles, what bone proteins and what immunological proteins, what host response proteins are altered in Staph aureus infection with the parent strain versus the SARA mutant. And you can see that the wild type strain, they all cluster together when uninfected shams and the SARA mutant basically all cluster with each other and are essentially indistinguishable. So we are completely reversing all the changes in these 119 proteins, host proteins, by mutation of SARA. And then finally, again, this is data from yesterday, but this I put in knowing who's following me. My friend and colleague, Dr. Killian, is going to talk about the immunological response in these infections. But here's these 119 proteins and assigning biological processes to them. And if you just go down this list, all of these are host defense response. All of them, response to biotic stimulus, regulation of immune response, regulation of innate immune response. Now, we haven't dug deep in the weeds on this data because I just got it yesterday, but I think it's really fascinating and informative. and something that warrants our attention moving forward. So my timer's going off. I don't have disclosures. I do have funding. These are my funding agencies. And then in the hope, Dave showed a slide that I actually, with an Einstein quote, but corrected me. I always thought it was Einstein, but apparently it wasn't. So I have to take the bumper sticker off my truck. But I'll close with this quote that I think is Einstein, that we all know light travels faster than sound. That's why certain people appear bright until you hear them speak. And with that, I will end my session and thank you for your time.
Thank you very much for a fantastic presentation. And you've already introduced our next speaker. So Tammy Killian is going to be speaking next on Staphylococcus aureus biofilm promoting leukocyte dysfunction during orthopedic infection. She's a professor of pathology in the Department of Pathology and Microbiology at the University of Nebraska, and already introduced. So thanks so much.
Okay, thank you. I want to thank Amber, Noreen, and Antonio for giving me this opportunity to present today. And also thank Mark for a great introduction to biofilm, and also Dr. Granger for a great overview of the immune system in response to orthopedic infection. So my laboratory is very interested in the interface between biofilm development and the innate immune response, which I'll be focusing on today. So we have several models of biofilm infection in the laboratory. Some that I won't be talking about obviously today are a cranial bone flap infection, where when people have craniotomy for brain tumor surgery or epilepsy surgery, a biofilm can form when that piece of bone is placed back in the skull. Here's a scanning EM from a patient with a Staph aureus infection on that bone flap. We also have a catheter-associated infection, but for today, I'm going to be talking about findings from an orthopedic model. I will mention, and here's some papers that we've published on the model, that we have looked at human prosthetic joint infection and have seen that a large majority of the findings that we see in our mouse model are recapitulated during human infection. So we feel it's a pretty good model to explore different immune evasive mechanisms. So this model includes a titanium K-wire that's inserted into the intramedullary canal, and we introduce just 1,000 bacteria directly into the joint. This causes a chronic infection in the tissue over a period of 1 month, the associated joint tissue, as well as on the femur and the implant. So Dr. Granger showed kind of an overview of a balance here between a pro and anti-inflammatory kind of environment. And my laboratory has been very interested in trying to understand why the anti-inflammatory mechanisms tend to win out. So during prosthetic joint infection, we do not see a lot of evidence for pro-inflammatory tone in terms of traditional immune effectors that would be associated with that response. Instead, we kind of see that the scale is tipped in balance of an anti-inflammatory type response. I'm not going to have time today to tell you about a major cell type we study in my laboratory, which is a myeloid-derived suppressor cell. We've also recently published a paper looking at epigenetic changes and Staph aureus metabolites that cause epigenetic changes in immune cells. We're also looking a lot at immunometabolism, and we're also looking at what the bacteria is doing to drive these processes. But today I'm going to focus on macrophage polarization during these infections and how they are skewed towards a more anti-inflammatory state. So as Dr. Granger mentioned, this is really not a true black or white scenario in terms of the way a macrophage responds. There is a gradation and plasticity in that response as well. But in general, if you have a macrophage that's exposed to a more traditional pro-inflammatory cytokine milieu typified by cytokines such as TNF or interferon gamma, these are typically thought to be elicited during a planktonic infection by staph caused from an abscess or sepsis. You then would have a macrophage that is primed to be pro-inflammatory, upregulate many pro-inflammatory cytokines, reactive nitrogen and oxygen species, and bacterial clearance. However, in a situation where we have biofilm formation, instead, these macrophages receive different cues. And this causes them to upregulate and express molecules that are associated with a wound healing response. So an increase in arginase, as well as anti-inflammatory cytokines such as IL-10, and this is important for bacterial and biofilm persistence. So we know over the years, a lot of work from my lab as well as some other laboratories, that Staph aureus biofilm evade recognition by classical antibacterial pathways that would be associated with a more planktonic infection. This includes evading toll-like receptor recognition. We've shown that with both toll-like receptor 2 and toll-like receptor 9. This is really important because the biofilm matrix has a lot of these molecules that are recognized by toll-like receptors. However, the biofilm somehow, and we really don't understand how this occurs, is able to circumvent that process. They also interfere with macrophage phagocytosis. Obviously, biofilm produce toxins to kill these cells. And as I already mentioned, they skew macrophages towards an anti-inflammatory profile by inhibiting pro-inflammatory cytokine and chemokine expression. They inhibit the infiltration of macrophages to the side of the biofilm and promote a fibrotic response. So here is some data from an earlier paper looking at a macrophage marker, IBA1, along with arginase 1, which is this prototypical kind of anti-inflammatory marker. And you can see here when we merge the two signals that there are a lot of macrophages in the vicinity, but importantly, this is where the biofilm is, indicated by this asterisk, so that the cells are not invading and expressing a lot of this anti-inflammatory molecule, suggesting they have anti-inflammatory tone. In addition, when we look to see where these cells migrate, here is a DAPI stain showing where all the cellular nuclei are. You can see here, this is the interface of where the biofilm is. You can see that there are cells there. However, they are not macrophages. So the macrophages are hanging out here at a distance away from the biofilm surface. So their ability to invade and actually contact that structure is inhibited. In addition, we know that if biofilm is not able to promote phagocytosis. So here we're looking at planktonic Staph aureus, shown here, the bacteria are in green. The macrophages have been labeled with cell tracker blue, so they're blue. So you can see here when macrophages are exposed to planktonic staph, this is no surprise. They are easily phagocytosed by these cells. However, if we look at biofilm, this is an orthogonal view, side view, as well as a top-down view. You can see all of these blue macrophages have no intracellular bacteria associated with them. So this is one way that they evade macrophage activity. So a new area that we're getting into in my laboratory is actually immunometabolism. So it's known that when a macrophage is exposed to either kind of a pro-inflammatory environment or cytokines that are more anti-inflammatory polarized, that this leads to specific metabolic changes inside the macrophage that are directly linked to its effector function. So if we look at a pro-inflammatory macrophage that's treated, exposed to tumor necrosis factor alpha or interferon gamma, these macrophages will have a very large surge in glycolysis, and they will have a reduction in their oxidative phosphorylation activity. Now, you may wonder, well, why is this happening? Because glycolysis is not a great way to generate ATP as opposed to oxidative phosphorylation. Glycolysis gives you 2 ATP, oxphos gives you 32. So the idea here is that when glycolysis occurs, you have the production of many metabolic intermediates that are essential for the high levels of protein biosynthesis and effector functions that these pro-inflammatory macrophages need. So glycolysis and all those glycolytic intermediates fuel things such as lipid production, protein production, and all of the molecules that help this pro-inflammatory macrophage exert its functions. However, a macrophage that is prone to be more anti-inflammatory has an increase in mitochondrial respiration because these are not as metabolically active and producing all of these effector molecules that are needed to kill bacteria. So our question was, do biofilm-associated monocytes in macrophages, are they more biased towards an oxidative phosphorylation phenotype? So here you can see we're measuring, this is a flow cytometry assay where we co-culture macrophages with Staph aureus biofilm, or just look at the cells by themselves. And you can see following co-culture, we have a significant increase in oxidative activity in these cells and not a very big increase in glycolysis. We're also looking at MDSCs and neutrophils here, but for the purposes of the talk here, we're just focusing on the macrophage response. So we do know, and this we've also seen this in vivo in the mouse model of prosthetic joint infection. When we take out macrophages from the site of the biofilm infection, they also have this increase in oxidative tone. So what we wanted to know is, could we target that metabolic activity to try to promote biofilm clearance? So here we used oligomycin. This is a drug that inhibits ATP synthase, which is the last step of the mitochondrial electron transport chain. And here you can see, if we look at a pro-inflammatory cytokine, if we add this inhibitor, we see more of a pro-inflammatory tone and a reduced anti-inflammatory marker. This is that arginase 1 again, that it was expressed in these kind of anti-inflammatory polarized cells. So the drug is doing what it should. And so we developed, in collaboration with investigators at UNMC, a nanoparticle formulation where we conjugated the nanoparticles to tufstin to target those two macrophages. This is a portion of the IgG fragment. And we labeled these with Cy5 so that we could see them at the site of prosthetic joint infection in our mouse model by IVUS. So when we delivered these nanoparticles, and I should say that these were delivered after the infection had taken place. So we waited until 1 week after infection to deliver the nanoparticles. So we know at that point a mature biofilm had formed in vivo. When we pull out the monocytes and look at their gene expression profile, we can see that when these oligomycin nanoparticles were delivered, we would shut down the electron transport chain and we would hopefully skew the cells to a more pro-inflammatory state. And that's actually what we see. So we see a reduction in the expression of many anti-inflammatory markers and an increase in pro-inflammatory gene expression. And so then the final thing that we did, again, this is an established biofilm, we gave the nanoparticles 1 week after infection, and then gave concurrent antibiotic treatment. The idea here is that if we could metabolically reprogram these monocytes, we envisioned that we might be able to promote maybe and start to disperse the biofilm so that it would be better amenable to antibiotic activity. So here I'm just highlighting the important ones here in the red squares. This is the, I guess I'll go back. These are the empty nanoparticles. And here are our oligomycin-only nanoparticles. So even if we just reprogram those monocytes to be more pro-inflammatory, we can see a significant reduction in bacterial burden. And again, this is an established biofilm. However, if we treat animals with just systemic antibiotic, we do see a bit of a dip here because in these experiments, we gave a pretty high dose of antibiotic. But what you can appreciate is when we do that, when we give systemic antibiotic, along with our ability to reprogram these monocytes and macrophages, now the bacteria are below the limit of detection. So basically, by metabolically reprogramming cells at the site of infection, we believe that we could potentially start to transform and awaken that biofilm that you heard Dr. Granger talk about to be more amenable to systemic antibiotics. We're also very interested in these MDSCs and metabolic changes that typify them as well. So just to wrap up then, again, we see a bias towards this anti-inflammatory profile, and studies are ongoing to really try to appreciate other mechanisms and importantly, how they all crosstalk and interface, because obviously this is a very complex situation. So finally, I'd like to thank the people in my laboratory that did the work. The nanoparticle work was done by a prior MD PhD student in the lab, and Courtney Heim is a senior scientist who's done a lot of the metabolic studies. And finally, I appreciate the funding from NIH and for all of you for your attention. Thank you.
Thank you very much for a great presentation. Without this next gentleman needs no introduction. Bias Bostrom is a fantastic orthopedic surgeon and researcher, and he's talking about the role of the immune system in a mouse model of periprosthetic joint infection, which he developed in his lab. He's the chief of adult reconstruction and joint replacement service and a professor of orthopedic surgery at the Hospital for Special Surgery. Thank you so much for joining us. Oh, I think you might be on mute.
Great. Sorry about that.
Good.
It's only been 11 and a half months of figuring out how to do this. So what I'd like to do is talk a little bit about our model. I'm not sure why my slides aren't advancing. Some disclosures, Dr. Vashka, oops, here we go, suddenly it's moving. Why don't we go ahead? So there are obviously some major questions in regards to the immune system and response to Staph aureus, and I think all the previous speakers have sort of hinted at that, both in terms of the innate immune response as well as the adaptive immune response. Despite having this robust immune system, we really can't clear these infections, especially Staph aureus. And our lab and Dr. Vashka's lab have had a lot of interest in the role of both innate and adaptive immune systems. Many of you guys know that we have developed a model of a articulating titanium implant that's 3D printed. It was initially developed for the use of in studying osseointegration, but Alberto Carlos, who I think is in the audience, thought, well, why don't we use this in terms of our interest in PJI? So this implant is titanium. We know that it forms biofilm, and we have used various doses of bacteria. Our workhorse has been Staph aureus using Zem36. Initially, we wanted to use it for the bioluminescence. We've never really gotten that to work very well, but we have continued to use it since we have so much predicate data in terms of the dosing. In terms of the study, we looked at early stage as well as late stages, and this is really work of two of our postdocs, one from Dr. Ivashka's lab, and the other in our laboratory, working closely with Dr. Carly, looking at this work. So initially we thought, what happens the first couple of days? I'm actually not moving that slides for some reason. That being said, our initial study was just looking at day 3. We looked at the implant itself. We've done that for many studies, looking at sonification, and then SEM through Karen Bentley up at the University of Rochester. But what's unique to this group of studies that I'm sharing today is that we not only did qPCR, but also flow cytometry. And really, this is a very powerful tool in terms of looking at these studies. So the initial results in this very, in some ways, a little bit of a pilot, we looked at the CFU counts. There we started with 10 to the 4th, and the substance studies we used 10 to the 3. And obviously, after 3 days, 2 out of those 3 had significant CFU counts. The others, we didn't measure the CFUs because they were done for scanning EM and histology. And this is what this looks like. You have the implant itself, and in high power, you can see not only the cocci of the bacteria, but also the biofilm itself. And this is very similar to our previous studies. In terms of histologic examination, here we have the non-infected legs, large number of granulocytes and PMNs in the canal itself, but no visible fibrotic tissue. In the infected leg, however, there's obviously abundant amount of purulent material. There's infiltrating neutrophils as well as monocytes around the implants. Obviously, at 3 days, there's no osseointegration, no ossification, and no fibrotic tissue. What makes this part of this study unique is that we also looked at what we call a local as well as a systemic response. And so we looked at local lymph nodes, those being the inguinal lymph nodes, and really didn't see a whole lot of difference between the right, which is obviously our infected, our implant side, which is always has been, and the left, which is the non-implant side. Obviously, they're different, but between the infected and non-infected, the local response at 3 days was not all that different. However, a more systemic response, which is where the periiliac lymph nodes, we did see a difference in the number of live cells. Looking at the myeloid population, there certainly looked like there was a trend of increasing neutrophils in the infected animals, both in terms of neutrophils, not so much macrophages and dendritic cells. And then obviously the IL-6 and IL-1 beta showed some differences in the, but not anything as dramatic as we had expected. In terms of the local immune response, these are the inguinal nodes. There were changes in CD3 positive cells, but the Tregs and T follicular helper cells did not show a difference, and nor did the CD4s. In terms of the systemic myeloid immune response, the neutrophils were obviously much greater in the infected side, infected animals versus non-infected animals, as were the dendritic cells. Macrophages were not different. And again, CD4 in terms of the lymphatic lymphoid immune response was greater in the infected as opposed to the non-infected sides. So this first step, and again, this is early time points, we certainly looked like we were able to at least study the cells that were there in a meaningful way. And then we looked at, and there were obviously significantly higher numbers of activated T cells in the para-iliac lymph nodes. Those are more of a systemic response. And looks like there was an induction of some dendritic cells. So obviously we thought we'd have to investigate this further. And that's why we looked at later stages. And in this group of animals, we really looked in terms of the tissue flow, we looked at 7 and 35 days, but we did a much more systemic approach as before as well, looking at 7 and 35 days. You can see the serum amyloid at 7 days is much greater. It seems to taper down, but still very elevated at 35 days. We looked at histology at not only 7 and 35 days, but points in between. And thanks to our pathology colleagues, we really have a fairly tight hold on the inflammation in the model is very clear, much greater in the infected, as you can see in panel C. Necrosis also greater, and the fibrotic response initially greater in the non-infected, but later on greater in the infected. Obviously, osseointegration takes a hit when the infection, especially at 35 days, where our histopathologic score for osseointegration, which we validated in other studies, really there is no osseointegration. And obviously, in panel C, you can see the inflammatory process going on. We did do looking at both innate and adaptive immune response, and obviously the innate response with IL-6 and other markers, TNF, were greater at 35 at both the early and late time points, and similarly adaptive responses, CD19 at 35 days was evident, not so much at the early time points, not surprisingly. Here's some of the flow data, which I'll share for the next few minutes. Obviously, happy to discuss each one of these panels, but we could probably spend my allotted 13 minutes on one panel alone. But needless to say, the innate immune response is dominated in both early and late stages, so both 7 and 35 days. You see elevation of cells that are predominantly associated with the innate immune response. And the adaptive immune response are elevated at late stages with flow, most specifically CD8 positive cells, as well as B220 positive cells. There were trends in CD3 and CD4 positive cells, but that was not significant. And then in terms of other cell populations, in both the spleen and the bone marrow, you can see that they're elevated in both day 7 and day 35. And then I think, both Upneed and Nirupama, who did all this hard work, isolating these lymph nodes is very tedious, and but they did a spectacular job of looking at the draining lymph nodes, these periiliac nodes at day 35, and show both CD3 positive T cells, CD4 positive T cells, as well as CD8 positive T cells. Obviously, we're seeing and really are really only scratching the surface now in terms of what the response is in this model. It can get into the different flow parameters and gating protocols were designed so to look at both gamma delta T cells, as well as a number of subpopulation of T cells, and still relatively early in terms of analyzing this data in terms of what this really means. But I think it's important to realize that there are these subgroups of T cells that are involved in the healing process. In time, I think again, qPCR showed sustained levels of both pro-inflammatory cytokines as well as elevated levels of gamma delta Ts and B cells in chronic soft tissue. We obviously, the lymph nodes become activated after the acute infection, and Tregs remain high at the chronic stage of infection in the iliac lymph nodes, with CD4, CD8 T cells expression in this time point. A lot to cover, a lot of detail. Happy to answer questions during the Q&A section. This was the whole lab. This was literally about 2 weeks before we were shut down last March. And really, both Upneed and Nirupama and Alberto deserve a lot of credit for wonderful work. So thank you all.
Thank you for a fantastic presentation. Really appreciate it. Last but absolutely not least is Amber Jennings will be speaking about hide and seek. Are we reaching biofilm in bone? She is a professor of biomedical engineering at Memphis, and she is going to come speak to us about this. And I'm honored to have led this session with her, as well as Noreen Hickok.
Thank you. I'd like to say that these have been some excellent talks. I'm going to talk a little bit today about my experience trying to develop biomaterials and some of the problems that we have specifically with biofilm in bone. So my disclosures are at the ORS website. So a lot of the other speakers have talked about biofilm. Biofilm is definitely life-threatening. You know, it causes up to 80% of infections. This is an image, an SEM image that looks a lot like some of the other speakers' images of biofilm forming on a titanium pin that we had implanted into a mouse model. And, you know, we've all, you know, in this rig, you know, we know that biofilm is very tolerant of antimicrobials, but that's not the complete picture. And so in addition to all of the issues that the other speakers have talked about, you know, we try to overcome this by using a local delivery system. So, you know, the types of things that we've tried might be electrospun fibers, injectable pastes. We've even tried ways to directly coat antibiotics onto a surface. We can use micro or nano beads. And then there are the good old standbys of PMMA and calcium sulfate pellets that are loaded with antimicrobials. And what we always seem to find is that these things are great at preventing an infection, but not that great at treating infection. And this was also something that at the consensus meeting a few years ago, you know, the outcome, you know, it was decided that local antibiotic delivery is not yet where it needs to be effective, at least on its own by itself. So, and we also have challenges in the lab. So this is showing some of our data on our local drug delivery systems that we tried in an ex vivo bone model, thinking, okay, if we can replicate the tissue conditions as much as possible, and we're going to try out our local delivery systems. And we were very excited that it seemed like we were making a dent in eradicating some of this biofilm. But when we tried the same thing in a model that resembled something like a two-stage revision, where the biofilm already had an opportunity to become established on the implant, then these local delivery systems were okay at getting to the actual implant biofilm. But when we checked the bone tissue, every single sample, we weren't really making much of a dent in the actual bone biofilm. So, what is it about bone that makes it so prone to infection? And so this is, particularly a problem for periprosthetic joint infection, as we've heard. Also, complex musculoskeletal trauma, like this injury involving soft tissue and potentially bone. And also, in areas where it's unavoidable to have, contaminating microorganisms directly near an orthopedic implant, like in dentistry. And so, we've heard also a lot about why particularly Staph aureus becomes the dominant pathogen in some of these situations. So this is from an excellent review article, and I think I saw soon to be Dr. Masters in the participants, and she's in Eddie Schwartz's lab. And so these images kind of represent some of, one of the major problems, and that's that the osteocanalicular network of the bone can be invaded by Staph aureus. And so this TEM image shows that what we know of as, a relatively non-motile bacteria actually inching its way, crawling through this narrow canalicular network. And when it does this, then it is shielded from these immune cells and are not really cleared by it. And so if you are interested in learning more about this, I would encourage you to go and find this other symposium on the ORS platform, Challenges and Opportunities in Musculoskeletal Infections. And Eddie Schwartz gives a great, more detailed explanation of what might be going on here. And then Antonia and Jeremy Gilbert and Bingyun Lee also have fantastic talks too. Another important aspect of this is that staph can actually hide within an abscess. So, this abscess can then shield Staph aureus from immune cells that could clear it. So, here's some examples from, this, also occurs in both bone and soft tissue. So, here is an infected model. This is a rabbit radius that's been infected. And, we have, used a local delivery system. And so we see these abscesses form in the bone and in soft tissue. But when we look at the gram stain for similar, for those same sections, then we see very, very little of that gram stained staph, but it is there. It's in these staphylococcal abscess communities. Another potential problem, and this is from a paper by Noreen Hickok, is that staph can form aggregates in synovial fluid, so even outside of the bone. So this is showing that, compared to normal growth media, the staph here is alive and growing and definitely could cause infection, but it's in these aggregates that are also more tolerant to antimicrobials. Another thing that Bingyun brought up, and these are images from Bingyun Lee in his paper, is that staph can invade the cells. And so this shows that they're actually inside the cells. And even more scary is that after they enter the cells, then they can become persisters and they can enter a semi-dormant state, and they can hang out there for months or years or decades and then reactivate infection at some later point. And this is particularly prevalent for small colony variants of Staph aureus. So, yet another problem is that we tend to overuse antibiotics, and I'm guilty myself of, designing local delivery systems to, hit things with a hammer. But the more we use antibiotics, then the more strains become resistant. We also last year heard a great talk about how these antibiotics can also affect the gut microbiome, and that is intricately linked with bone health and could actually be hurting us rather than helping us. So when I think of all of the things that we need to come up with, as innovations and biomaterials and therapies to treat infections, so some of the hurdles we have to jump through are, we need to have something that gets into the osteocanalicular network. We need something that also reaches the staph that's hidden away within abscesses. While we're at it, we need to also make sure that the staphylococcal aggregates that might be outside the bone but in the synovial fluid are eradicated. And then if we could design something that could also make sure to get into the bone cells and the macrophages without killing them, but kill the staph, then that would also be great. If we could prevent these bacteria from forming persister cells within the tissue. And while we're at it, if we could just go ahead and find something that is not one of these traditional antimicrobials that we'd like to reserve for other uses and make sure that they're effective for the longest time possible, then that turns out to be a very, very tall order. So with that, I will conclude and ask for questions, but not only questions, but also ideas and solutions. And so remember to click on the PDF that's uploaded in the chat, and I think there are also some links pasted within there, so that we can jump off of this Zoom session and discuss all of these wonderful talks a little bit more.
I'd like to thank all the speakers, and please do what Amber is asking, which is to jump on over to this Zoom chat. We'll resume in about 5 minutes, and I'm sure there's going to be a lot of questions for all the speakers, and we look forward to a great discussion. See you over there. Please be sure to either download the link or the PDF, because as soon as you leave, that chat is gone. Thank you.